My name's Molly Henderson. I'm a student at the Loomis Chafee School in Windsor, Connecticut. I'm a senior there. And I'm super excited to get to be here and to meet so many other young women who are interested in science and STEM. And today I'll be presenting the research I have been working on studying the genetics of stem cells. So my school, Loomis Chafee, has partnered with Dr. Christine Fink's lab at the University of Connecticut in order to help them better understand how stem cells work and their applications in regenerative medicine. Dr. Fink's lab is currently working on regenerative medicine for a condition called esophageal atresia. Esophageal atresia is a very serious condition in infants where there is a break, as you can see, in the esophagus right there. This is a really serious issue. However, current methods of treatment are not ideal. One method of treatment is to pull the stomach up higher in the chest cavity and sew the two ends of the esophagus together. However, due to the shortened esophagus, this can cause a lot of acid reflux and discomfort. Another solution to this problem is to use a piece of the patient's own intestine to bridge the gap. However, the muscles of the intestine are not suited to swallowing food as the esophagus is. And so this can lead to difficulties with swallowing food. And additionally, the high acidity of the stomach really hurts the fragile intestinal lining. So Dr. Fink's lab is working on a solution to this problem that promises to reduce post-surgical complications like immune response by growing the new esophageal tissue on a piece of synthetic tubing called a scaffold using the patient's own stem cells. But we don't know how the stem cells know to go to the scaffold and repair the atresia. And this is ex vivo, but it can also be done, the actual procedure would be done in vivo. So the little piece of tubing that you can't really see here would be placed inside the patient and the cells would grow on it. We've had some success with this in pigs. However, we haven't gotten to human trials yet because it's very experimental so far. So stem cells are unspecialized cells that have the potential to become any type of cell in the body. Now, this means that they have the capacity to control growth and repair tissues. However, researching the genetics of stem cells could help us to understand why they are able to go to the scaffold and repair the atresia on it. Here are pictures of the a picture I took of our stem cells that we're working with in our lab through a microscope. These are stem cells that were harvested from one of the esophageal atresia patients at UConn. And I chose to look specifically at a gene called IGF-1. IGF-1, or insulin-like growth factor 1, is a protein that works a lot like insulin in the body, as the name would suggest. It is in charge of some of the anabolic processes that store small carbohydrates as larger molecules for later use. Additionally, IGF-1 is very important to childhood growth, and furthermore, research suggests that periods of fasting or low glucose levels can lead to can stimulate stem cells to repair tissues and grow them as well. So my hypothesis was that IGF-1 is expressed in stem cells and that the expression of IGF-1 increases under low glucose conditions. To test this theory, I first grew and harvested the stem cells. I then broke open the cells to extract the RNA. I decided to look at the RNA because RNA includes only the cells that are being only the genes that are being expressed in a cell. So the IGF-1 gene is encoded in the DNA of every cell in your body. And so in order to know if it's being expressed, if the protein is being created, I needed to look at the RNA. I then reverse transcribed the RNA into DNA because DNA is a much more stable molecule and is easier to work with. I then used a process called PCR, or polymerase chain reaction, which is a method by which scientists are able to replicate a specific section of DNA that they want to study artificially in the lab. The methods used in PCR are very similar to those that your body employs during DNA replication. So I now had a lot of copies of just the IGF-1 gene that enabled me to study the product through gel electrophoresis. Gel electrophoresis is a method by which scientists put DNA samples that they want to study in a piece of firm gel that kind of feels like hard jello. When an electric current is passed through the gel, the DNA, due to its negative charge, flows towards the positive electrode. Now, smaller pieces of DNA, so shorter in length, travel faster through the gel because it's easier for them to fit through. This allows for samples of DNA to be separated by length. So, therefore, since all the copies of the IGF-1 gene are the same length, they'll form a little band, as you can see in red there, at the appropriate place on the gel. As you can see here, this is a picture of my gel that I took, and indicated in red, you can see the band that has formed in the IGF-1. This indicates that IGF-1 is expressed in stem cells, and the first part of my hypothesis is correct. However, the second part of my hypothesis is that expression levels increase under low glucose conditions. To test, oh my. To test this hypothesis, I performed the same experiment, but with two sets of cells, one grown in high glucose media or high glucose conditions, and one grown in low glucose media. So then I harvested the cells, extracted the RNA, reverse transcribed, and performed a PCR. And when I studied it in the gel, through gel electrophoresis, I found that IGF-1 was expressed in both high and low glucose media. So the next step in my experiment is to quantify how much IGF-1 is being expressed in these stem cells. So in order to do this, I will perform the experiment extracting the RNA and replicating it again, and then I'll use a method called qPCR, or quantitative PCR. This is a method by which scientists are able to quantify how much of a specific gene they have in a sample by putting a fluorescent tag on the gene, so that when it is replicated during PCR, a sensor is able to pick up on the fluorescence. In conclusion, IGF-1 is expressed in stem cells, and it is expressed in both high and low glucose conditions. However, more research needs to be done concerning the second part of my hypothesis, and I need to quantify the expression levels. Now, excuse me. This and more information about the genetics of stem cells will hopefully facilitate more advancements in regenerative medicine, like those in esophageal atresia. I would like to thank everyone we worked with at UConn, including and especially Dr. Christine Fink and her senior scientist, Mr. Todd Jensen. Thank you to my classmates, thank you to Lead Like a Girl, and most importantly, thank you to my teacher, Dr. Durace, for all her support and guidance. Finally, thank you all for your attention. I'm so glad that I got to share my research with you. Always remember that you can do amazing things. You have the ability to do great things, and to never let anything get in the way of following your dreams. If you have any further questions, please do not be afraid to ask.
Very intriguing. So what do you think the next steps are of your research? Where would you go next?
So for my research, I want to quantify the expression levels, and then from there, I really want to use RNAi or RNA interference to stop the IGF-1 gene from being made, and so that we can see really the effect that the IGF-1 has on the stem cells. Or I could tag the IGF-1 gene with a fluorescent tag, and that will make it glow under a microscope. And so then I can use that to actually track the protein and like see what it does and how it functions in the stem cells, which is really exciting.
It will feedback into the work that the doctor's doing about this.
Yeah, I think our hope is that the understanding more about the genetics and how they work, how stem cells work, and why stem cells are able to do these things and recognize the scaffold will be understood by the genetics. So if we know more about how they do things or why they do things, we can use them more effectively. Okay, yes.